Хазайлмын уянгаа байна. Өнөөдөр Fernando бит хоёр I'm going to introduce you. Fernando болохоор манай лабын Postdoc. Би бас Postdoc. Бит хоёр өнөөдөр нийлээд primer яаж дизайн хийх вэ, яаж захиалах вэ гэдэг талаар харуулах гэж байна. Тэгээд Fernando болохоор энэ тал дээр мэрэгчлэлтэн байнга захиалж энэ тал molecular biology work-саа хийдэг байхгүй. Би чинь болохоор чинь амьтны туршилт хийдэг шүү дээ. Тэгээд Fernando аль чи тэрийг заагадаг. Намайг тэгээ магистрийн оюутан гэж бодоод тэгээд яг өөрөө PI professor гэж бодоод тэгээд заагаараа гэлсэн баг шүү. За. Let's start. Бид хоёр тэгээд англиар бид хоёр англи хэл тийм амар сайн биш. Тэгээд англиар ямарчсан гэд яриад явна. За. Тэгээд ингийн англи дээр ярина. I just told them we're going to use our stock English. Please understand. Okay, let's get started. And first, we need to open Chrome. We need to decrease our cells. Okay, here. Okay, he's going to explain what is the primer first. And then, yeah, if I have a question, I'll ask Fernando for you instead of for you guys. Okay, please pretend you are the PI. I'm master degree student.
Okay,
Yes.
well, first of all, I think it's very important to explain that for a PCR, it's very important to have two primers, the forward primer and the reverse primer. The forward primer will bind to the 3' to 5' strand of the DNA, and the reverse primer will bind to the 5' to 3' to the DNA to create the product, like the amplified product. A PCR consists basically in 3 steps: the denaturation, annealing, and extension. Denaturation exactly basically is like separate the 2 strands of the DNA. Then the annealing means bind the primers in each strand of the DNA, and extension is the step in which the polymerase will use the DNTPs and the primers to create a new strand. And this has to be repeated like many cycles, like for example 30 or 35 cycles to create your product, your amplified product.
Okay, so I have a question. Please explain what is the 3' or 5'. For that, I prepared some DNA structure picture. Can you explain it?
Yes. So basically, as you know, the DNA consists in 2 strands: the 5' to 3' strand and the 3' to 5' strand. These 2 are the same, but they are complementary to each other. So as you know, also the DNA consists in 4 type of nucleotides. They are adenine, thymine, that will create a bind, and guanine and cytosine that also will create a bind. So
Yeah. Also, this adenine thymine bond called this hydrogen bond, 2 hydrogen bond. Guanine, cytosine is 3 hydrogen bond. That's why it's guanine cytosine is a more strict bonding it's made. So our primer should consist 40 to 60% GC
exactly
nucleotide, right?
Exactly.
Yeah. Also, I want to add something here. The DNA consists with the deoxyribosal, and on the 5' on the 5' it has a phosphate group. Also, 3' it has a hydroxyl group. So DNA extension always goes to the 5 to 3' like this, right? So then he's going to explain how to generate primer using this. What is NCBI?
So first of all, I think it's very important to mention that the design of your primer depends on the objective that you want to do. For example, if you want to design primers for qPCR, you have to have different considerations. If you want to design primers to detect, for example, a virus. You have to have other considerations. Let's pretend in this moment that we are designing primers to detect a specific mutation that we created, for example, using CRISPR or genomic edition or whatever.
Yeah.
So also, I think it's very important to mention that I always like to design my own primers to check all the process, to check all the sequence. But I think it's very important to say that there are a lot of papers that already have reported different primers.
Yes.
So if you have the opportunity to read a paper and they are studying a gene that you are studying in this moment, they reported the primers for some reason, so you can use it, and that's easy. But I will recommend to make this exercise of creating your own primers since the beginning.
Okay. Please show how to.
So
generate.
this is NCBI. You probably already know this site. It contains all the sequences of different genes. So let's click in gene and let's search for a very important gene called FASN.
FASN, okay.
Let's click in search. And then here you have different species, Mus musculus, Homo sapiens.
It's a human, okay.
Let's click human. And if we go down, we will have the sequences for the messenger RNA, and also you will have the complete genomic sequence of the complete gene in FASTA format. So it's important to click here in FASTA.
FASTA, okay.
And you will have, after some time, the complete sequence.
Yeah.
So, for example, if you want to detect a mutation that you created with CRISPR, it's important to take, for the designing of your primers, some nucleotides before and after of the site of your mutation. So let's pretend that our mutation, I don't know, is, for example, in here, in some region here. So let's take all this region of the gene. Like this is enough.
Okay. And copy.
Copy, and then there are different websites to design your primers. I like to use, for example, Primer Blast. It's only necessary to Google for Primer Blast, and then here in Interact section you can enter the number, the specific number of the gene, but you can also paste the complete sequence. So let's paste the complete sequence in here. You have also here the opportunity to introduce your own primers in here. Check the product size. I mean, if you need a specific product size, let's pretend in this moment that we need, I don't know, for example, 400 to 1000 of nucleotides.
Because it has a mutation, sorry for interruption. Because it has a mutation, so we creating a bigger one.
Exactly.
If it has no mutation, how long we should choose the PCR product size?
Again, it depends on your specific objective.
Usually, common.
Maybe 200. I mean, if you only want to detect a gene in a specific sample, 200, 300 is more than enough.
How about like housekeeping gene, CAPTH or?
The same. 200.
Same 200.
Yeah.
Okay.
But for example, if you are designing primers for qPCR, sometimes it's better to have a small product size, like for example 190 nucleotides. Yes. So we have other parameters. Let's here in the database, let's select NT, and we are searching for human. So it's very important to have here human and show results in a new window and get primers. And this will take maybe 3 to 4 minutes.
Okay.
And we will have all the results with all the different characteristics, like for example the product size, the melting temperature, what else? The GC content that you at the beginning said this was very important, because the GC content depends the denaturation temperature, because they have 3 hydrogen bonds. So it's very important to check all this information.
Okay. Oh, during this waiting time. Oh, it's already done.
So let's click in the first one.
Okay, why? Just because of force?
Because the identity is 100%, so it's probably the gene.
Okay. So it's unique. Huh? Oh, during this minute we can show some your paper.
Yes, but this is a read for B, so it's different.
It's different.
Search
For example, right?
But I have other paper FASN.
Oh, really?
Yes.
Oh, so.
But I don't know if it's public.
Then we can explain.
Okay.
Instead of creating primer as a new one, no one used one, we could use, as Fernando said, we could use the NCBI, but it's a, as Fernando said, is it very good choice if that primer somebody already used it and published it, we could copy it, right? Like this copy. This is Fernando's paper, by the way. See, Fernando Gonzalez Salinas. So we could use already published paper. We could copy primers and just authors, right? Okay.
Yeah, you can literally only like copy and paste the primers in your favorite company like Thermo Fisher or IDT and order it. So let's return to primer blast. It needs maybe 2 or 3 minutes more.
Okay. Yeah, I have one question during this waiting time. Why is the melting temperature so important? And if it is too high melting temperature, what will happen? Also, how could we stop that extension? Okay.
So the melting temperature is very important because on this temperature depends the annealing temperature, that is the temperature that you have to program in your PCR machine for separating your DNA, your two strands of the DNA, and your primer and your DNA. Exactly. So, for example, if your annealing temperature is lower than the necessary temperature that you have to program in your PCR machine, you probably will have some inspecificities. Like, for example, the primers will start binding in another places in your DNA, producing more PCR products than the PCR products that you want.
Yes.
And if, for example, your annealing temperature is higher, your primers will never bind to your DNA. So it's very important to check all this information and set your temperatures in your PCR machine in a correct way.
Usually, denaturation temperature is always 95 degrees.
Yeah, it's a high temperature.
Annealing temperature is 50.
58, 60.
Yeah, 55 to 65. And yeah, extension, if we do not adjust the time, extension never stop, right? So it's always 30 seconds or something?
30 seconds, something like that, yeah. But for example, if you set your extension temperature in 5 minutes, the polymerase will continue. I mean, they have a life, but they will continue. So the extension temperature also depends on the product size. So for example, your product size is 50 nucleotides or 100 nucleotides, maybe 15 seconds is enough. For that reason, it's very important to take that into consideration.
Yeah, just 15 seconds, okay. And then all cycle usually 35 seconds. 35. Repeating, right? And then we have to adjust the end of the cycle 4 degree.
Just to preserve your sample, like a fridge temperature. It's like you are telling the PCR machine, please store my sample in a correct way.
Yeah, PCR temperature cycle images. Yeah, for example, here. It's a denaturation, it's a DNA separation, and annealing, an extension, just 1 minute, and like 35 times. Yeah. We have to adjust that machine, right? Like this. Okay, let's check this one.
So here the website is showing like different pair of primers. You can select the pair of primers that is better for you dependent of the location. For example, if you have your mutation in here, it's better to use this primer, I mean the number 4. And here you have all your information. You have the primers. This website always will show you the
forward and reverse.
Exactly, forward and reverse, and in this format, the sequence in 5' to 3'.
Forward is always the region of interest start. Reverse primer is the region of interest is end. Right? So yeah, okay.
And your product size includes the primers, I mean the sequences. Then you have the length. Normally the length of the primers are 18 to 22. 20 is perfect.
It's nucleated. Okay.
The melting temperature is 60, so it's perfect. And the GC content is 55, which also is perfect.
Yes.
It's also very important to check the cells complementarity because, as you know, the primers maybe can have the ability to bind itself, creating secondary structures. So it's very important to check also this. And here you have all your primers, I mean your different possibilities. So you can select, for example, we set the primer number 4. We can go to Pero primer number 4.
Here it
and is this one. So the product size, the product length is 997. I think is good. And again, the melting temperature is near 60. GC content is 55. So we can be ready to start ordering these primers.
So please show how to order.
Yes, well, as I said, there are different companies, like for example IDT and Thermo Fisher. Here in the lab we both use Thermo Fisher.
Thermo Fisher, okay.
So we only need to copy, for example, first the forward primer, and here in the custom standard DNA oligos in Thermo Fisher, you can write fast N, that is the name of the gene, forward. The researcher name is Uyanga, and the oligo sequence we have to paste in here. Then synthesis scale is the concentration of your primers. 25 nanomoles is more than enough almost all the times. The purification, the most common is desalted, and here you have some options if you want to add some modifications to the 5' end or the 3' end. In this case, probably not necessary, and that's it. You can add, for example, another oligo. You can write fast and reverse. The researcher name is the same. We can return to primer glass to copy and paste your primer. Let's write here first a T, and that's it. I'm not sure if T, exactly. Okay. And same. Okay. Normally primers are very cheap in this moment, and that's it. Add to cart.
After our duration, it arrived. Then how to dilute it, and it should be RNA-free water or just tea? What is tea? Why it should be tea?
Is a buffer, if I am correct. Let's check, but I think it's 3 plus EDTA. So you can respond in TE buffer. I normally respond in water. Molecular biology grade is more than enough. And when you receive your primers, they will tell you the amount of water that you need to add to your powder to have like the suspension of primers. So when they arrive, you have to add the water and store at -20 degrees.
Okay,
And that's it. Proceed to checkout, and you can tell your lab manager to order.
Thank you, Fernando, for explaining and for teaching us how to order primer. And next time we're going to teach some new topic, right? So let's finish our video. And thank you for watching. And bye-bye.